Scientists studying a cousin of the bacteria responsible for tuberculosis and leprosy have discovered an enzyme that converts hydrogen into electricity, and they think it could be used to create a new clean source of energy, literally from thin air. The enzyme, which has been named HUC, is used by the bacterium Mycobacterium smegmatis to draw energy from atmospheric hydrogen, enabling it to survive in extreme nutrient poor environment. Now, by extracting and studying the enzyme, the researcher says they have found a new energy source that could be used to power a range of small portable electrical devices. They published their finding in the journal Nature. We imagine that a HUC containing power source could power a range of small portable devices using air, including biometric sensor, environmental monitor, digital clocks, and calculators or simple computers. When you provide HUC from more concentrated hydrogen, it produces more electrical current, which means you could use it in fuel cell to power more complex devices like smartwatch or smartphone, more portable complex computer, and possible even a car. M. smegmatis is a non-pathogenic, fast-growing bacteria often used in the lab to study the cell wall structure of its close, disease-causing relative Mycobacterium tuberculosis, commonly found in soil all over the world. M. smegmatis has long been known to convert trace hydrogen in the air into energy. In this way, the microbe can survive is the toughest environment, including Antarctic soil, volcanic craters, and the deep ocean where a little other fuel can be found. But until now, how M. smegmatis did this was a pervading mystery. To investigate the chemistry behind M. smegmatis' shocking ability, the scientists first isolated the HUC enzyme responsible for the process using chromatography, a lab technique which enables scientists to separate the component of a mixture. Then they investigate the enzyme atomic structure with cytoelectron microscopy, a technique that won its creator. By beaming electron onto a frozen sample of HUC that was gathered from M. smegmatis, the researcher mapped out the enzyme atomic structure and the electrical pathways it used to carry the electron so they form a current. The team discovered that its center, HUC, has a structure called an active site that contains charged ions of nickel and iron. Once hydrogen molecule made up of 2 proton and 2 electron enter the active site, they become trapped between the nickel and iron ions and get stripped of the electron. The enzyme then sends these electrons along in the flowing stream to generate current. The electrons are absorbed by HUC and transferred to the surface of HUC by a molecule wire formed by a cluster of iron and sulfur ion. If we immobilize HUC on an electrode, the electron can enter an electrical circuit from the enzyme surface and generate current. Further experiment revealed that the isolated HUC enzyme can be stored for prolonged period, that it survives being frozen or heated up to 80 degrees Celsius. Then it can consume hydrogen at concentration at minuscules at 0.00005% of that found in the air we breathe. These attributes, alongside the microbe ubiquity and ability to be easily grown, could make the enzyme an ideal candidate for a power source in organic batteries. HUC can extract energy from hydrogen in the air, which is effectively limitless. The amount of electricity that can generate from the low concentration of hydrogen in the air will be modest. This will limit the application of HUC in this content of device that requires a small but sustained amount of power. A complementary use of HUC would be in fuel cell, where a higher concentration of hydrogen is provided. Mars ain't the kind of place to raise your kids. In fact, it's cold as hell. But new research suggests that Martian chill could allow bacteria to survive for up to 280 million years below the planet's surface. The finding raises hope that trace of ancient life or even viable organism in suspended animation could be found on the red planet someday. In the study, scientists found that an earth bacterium, Deinococcus radiodurans, is so resistant to radiation that it can handle the equivalent of 280 million years of the radiation present 33 feet 10 meters below the Martian surface. The plucky little microorganism, which has been found thriving in nuclear reactor on Earth, could even last 1.5 million years on the Martian surface, which is constantly bombarded with cosmic and solar radiation. The key to this survival is Mars' dry, cold environment. When desiccated and frozen to minus 110 degree 79 degrees Celsius, the temperature of dry ice and the higher latitude region of Mars become phenomenally astronomically radiation resistant. Radiodurans has been known to be a champ at rising radiation found in the human gut and in many other places on Earth and has even survived for years in the vacuum of space. The new researcher, however, in the first attempt to test the bacteria upper limit of radiation resistance when it is desiccated state. Previously, scientists had found that the bacteria can withstand 25,000 grays of radiation when a liquid culture. For comparison, a dose of 5 gray would kill a human. Deradurans and the bombarded the bacteria with both gamma radiation and proton radiation, mimicking cosmic radiation from deep space and solar radiation from the sun. They found that dried and frozen deradurans could survive a mind-boggling 140,000 grays of radiation that equivalent to those from 1.5 million years on the Mars surface and 33 feet beneath the surface, whereas the only radiation is from the radioactive decay within rocks and minerals. The organisms survive irradiation in two ways study. First, they have multiple copies of their genome, providing a backup for any bits damaged by radiation. Second, they accumulate large amount of manganese antioxidants, which capture damaging molecule created by radiation. The capture of these molecules prevent damage to the protein that do DNA repair from the organism. The DNA is organized to be repaired, and these manganese antioxidants protect the protein that do the repair. Deodorants evolved on Earth where the atmosphere protects the planet and its organisms from the worst radiation. The bacteria probably evolved to withstand damage during dry period, and the radiation resistance is just side effect of the evolution. Any Martian bacteria would have had to evolve in the environment without that protection and likely would have had to evolve similar radiation resistant. Mars has not had widespread liquid water for 2 billion years, so even if ancient life did evolve there, 280 million years still too short to suggest that the planet hosts a plethora of bacteria just waiting to spring back to life. But, but because Mars has very thin atmosphere, meteorite rain onto the planet's surface regularly. The heat and liquid water released those impact could potentially warm up dormant bacteria in the surface and allow for a temporary flourishing of life. Even if this temporary oasis theory isn't true, the long-lasting potential of bacteria on Mars means that bits of piece of ancient life could still be present as traces in the rock. DNA and other signature of life could exist as fragment even if the organism were long dead.